Hi everyone, my name is Lindsay, and today we're going to be talking about mRNA translation and how it relates to the COVID-19 vaccine. mRNA, or messenger RNA, is a type of single-stranded nucleic acid, and it carries genetic information that codes for proteins in the body. mRNA is made via transcription from DNA and is then used to make polypeptides during translation. You've probably heard about the mRNA COVID vaccines, and this type of vaccination introduces synthetic mRNA into the body. Translation is the process by which mRNA is read and made into proteins composed of amino acids. There are 4 steps of translation: tRNA charging, initiation, elongation, and termination, and all of these take place in the cytoplasm of a cell. Because the mRNA vaccine codes for a special spike protein found on the surface of the virus, the body will begin translating the new mRNA to make its own spike proteins, and then the body can build up an immune response to these foreign molecules. In order for translation to occur, molecules called transfer RNA must be charged with their respective amino acids. Each tRNA has a series of 3 bases called an anticodon that corresponds to the amino acid it must carry, and they bind together using amino acyl synthase. Next, initiation occurs when a ribosome made of ribosomal RNA and a charged tRNA attached to the mRNA strand. The complex then moves along the 5' untranslated region of the strand and scans for the start codon, which indicates the point where translation will start. During elongation, the polypeptide chain is built. The ribosome moves along the mRNA in the 5' to 3' direction, reading the nucleotides 3 at a time. A charged tRNA will enter the ribosome and a peptide bond will form between the growing polypeptide chain and the new amino acid. tRNA molecules enter, shift, and exit the ribosome continuously until a polypeptide chain is fully formed. Finally, translation is terminated once a stop codon is reached. Instead of tRNA, a release factor will bind with the stop codon, signaling for the protein to be released. Then, the ribosome disassembles and separates from the mRNA. The section of mRNA beyond the stop codon is called the 3' untranslated region, but it does not contain any information for the target protein. To help understand translation, let's use a baking analogy. The mRNA strand is like a cake recipe that tells us what ingredients we're going to need and how to make the cake. The amino acids are analogous to the cake's ingredients because they make up the final product. Lastly, the cake is like the spike protein at the end of translation. We must first go out and get all the ingredients we need, much like the tRNA getting their amino acids. In order to get ready for the baking to begin, we prep the batter, much like initiation. We put the batter in the oven so that the cake is baked, which is like building the polypeptide during elongation. Finally, we terminate the baking by removing the cake from the oven, and it's ready to eat. In regards to COVID-19 and viruses in general, mutations are common and can lead to different strains. This can be problematic for vaccine efficacy depending on the type of mutation that occurs. Silent mutations are point mutations where the changed codon will code for the same amino acid as the non-mutated sequence. Because the amino acid is the exact same, the protein would not be altered, so these types of mutations would not really affect the mRNA vaccine. Missense mutations, however, code for different amino acids. Since this would ultimately change the makeup of the protein, it would affect the vaccine. In order to combat this, vaccine companies may need to alter the mRNA sequences they use in the future to match the specific mutations seen in the mutant strains, much like the one shown on the screen. Here's a list of my sources, and that concludes this video on mRNA translation and the COVID-19 vaccine. Thanks for watching.